Hi everyone. Today let's learn the topic antigens. So today's topic, our learning objectives will be first of all to define antigen. So we will be defining the antigen. Secondly, we will be studying regarding the factors influencing the immunogenicity, and third one will be we will be discussing regarding the biological classes of antigens. So after studying this topic, you will be able to define what an antigen is, what are the factors influencing the immunogenicity, and also the different biological classes of antigens. Now, you have studied in your previous classes that antigens are foreign particles, right? So, antigens are the substances or the foreign particles that can stimulate immune response. So, being said that, these antigens, they should be able to satisfy two immunological properties. One is the immunogenicity and the other one is the antigenicity. Now, what do you mean by this immunogenicity? Immunogenicity is the ability of an antigen to induce immune response in the body. So what are the two types of immune response in the body? One is the humoral mediated immune response and the cell mediated immune response, right? So the antigen should be able to induce either humoral mediated immune response or cell mediated immune response, or both humoral and cell mediated immune response. Being said that, when this antigen enters into the body, they come in contact with the B cell and it will be differentiated into effector B cells, that is the plasma cells, which produce the antibody, as you know, and the other one is the memory B cells. And if the antigen comes in contact with the T cell, they will get differentiated into effector T cells, that is T helper cell or T cytotoxic cell, and also will produce memory T cells. Now, the substance which induce specific immune response is known as immunogen. So they are known as immunogen rather than the antigen. So more specifically, we can call them as immunogen. Now coming to the second property, that is the antigenicity. So antigenicity means the ability of the antigen to combine specifically with final products of the above two responses. So what will be the final product of the above two responses? That is, when B cell comes in contact with the antigen, they will be producing effector B cells, through which they will be producing antibodies, right? So one of the product will be antibody, and the other one, when T cells come in contact with the antigen, they will be producing T helper cells or T cytotoxic cells, which will be having the T cell receptors. So these are the final products of the above two responses. In that case, the antigen should be able to combine specifically to these antibodies and or the T cell receptors. So such antigens are known as complete antigen. So such antigens are known as complete antigens, that is, which has the ability to induce the antibody formation and they have to react with the products. Now, all molecules having immunogenic properties always shows antigenicity, whereas the reverse is not true. That is, the molecules having antigenic property will not show immunogenic property. So keep in mind, children, that is, the molecules having immunogenic property shows antigenicity, whereas there are some molecules which shows antigenicity but they are not able to elicit immunogenic property. Okay. Now, when we take an antigen, there are small areas, okay, there are small regions on the surface of the antigens, probably some 4 or 5 amino acids, okay. So these areas are present on the antigen and they are capable of sensitizing. Actually, they are capable of sensitizing the T cell and B cells and react with the specific site of the T receptor or an antibody. So such an area is known as the epitope. So epitope is otherwise known as antigenic determinant, which is the smallest unit of antigenicity. So what you have to keep in mind is epitope is the smallest unit of antigenicity. It's nothing but it's a small area present on the antigen. Okay, it's a small area which is present on the antigen. So here you can see it's a very small area when compared to the antigen, which is present on the antigen and which is capable of sensitizing the T cell or B cells, and they'll be able to react with the specific site of T cell or the B cell receptor. And the specific site of the antibody that react with the corresponding epitope is known as the paratope. So when it comes to antigen, it is called as epitope, and when it comes to antibody, it is known as paratope. Now, these epitopes are grouped into two, or they are of two types. One is sequential epitope, or they are also known as linear epitope, and whereas the other one is known as non-sequential epitope or conformational epitope. Okay, so this sequential epitope is present as a single linear sequence of few amino acid residues, whereas the conformational epitope or the non-sequential epitope, they are found on the flexible region of the complex antigen having tertiary structures. Okay. Now, coming to, here we discussed regarding complete antigens, right? Now, there are few substances which cannot induce immune response but have got the antigenicity. So such substances are known as haptens. So keep this particular point in mind. So such substances are known as haptens. So what are haptens? Haptens are low molecular weight molecules which cannot induce immune response. So no immune response, but they have got antigenicity. Okay, they have got antigenicity, whereas they do not have they do not have the ability to induce immune response. Okay, and which means that they cannot activate helper T cells containing single epitope, and they can, but does it bring any reaction in the body? Yes, they can become immunogenic when combined with a larger protein molecule which is known as carrier molecule. So these haptens plus a larger molecule, that is a carrier molecule, can induce immune response. So keep in mind, haptens cannot induce immune response. These are low molecular weight substances which cannot induce immune response, but they can retain the antigenicity or immunological reactivity. Okay, whereas, and they cannot activate helper T cells containing single epitope, but they can become immunogenic when they combine with the larger protein molecule which is known as carrier molecule. Okay, so these hapten carrier complex, they are able to induce the immune response. Now, these haptens can be classified into two types. Okay, they can be classified into two types. One is a complex hapten and the other one is a simple hapten. So, as the name suggests, simple hapten, they have got only one epitope. Okay, they can bind to the antibody, and hapten antibody complex cannot be visualized. We cannot visualize this particular reaction. Okay, and whereas the second that is the second classification, which is the complex hapten, they contains two or more epitopes and they can react with specific antibody, and they can be visualized by precipitation reaction. You already know that precipitation reaction, you have studied regarding precipitation reaction, right? When an antigen and antibody combines, you can visualize them, right? And that is the precipitation reaction. So here you have studied two types of haptens, and they are simple haptens and complex haptens. Simple haptens contains only one epitope, and when they bind with the antibody, you cannot see the complex antigen antibody complex formed. It cannot be visualized. Whereas complex haptens, they contain two or more epitope and they can react with a specific antibody, and the reaction can be visualized in the form of precipitation reaction. Now, these antigens, based on the antigen-host relationship, you can classify them into different types. Okay, so that is, one is the self or autoantigen, which belongs to the host itself. So as you know, when you hear the name, that is self antigen or the autoantigen, it means that it belongs to the host itself, and hence it is not immunogenic. Okay, the host immune system does not react with its own antigen due to a principle known as immunological tolerance. You will study regarding that later. Okay, and the second one is non-self or the foreign antigen, regarding which we have already discussed, and that is actually immunogenic. So self antigen, it is non-immunogenic, whereas non-self antigen or the foreign antigen, it is immunogenic. Okay, now these antigen can also be classified into different types based on the phylogenetic distance to the host. Okay, that is based on the phylogenetic distance with the host. These foreign antigens, okay, not the other antigen, foreign antigens can be classified into different types. One is alloantigen, second one is isoantigen, third one is heteroantigen, and the fourth one is heterophile antigen. So you have got four different types of what? Four different types of foreign antigen. Okay, you have got four different types of foreign antigen. One is alloantigen. So these alloantigens are species specific. Okay, so all the organism, that is the tissue of the all organism, they contain a specific species specific antigen. So this species specific antigen is known as alloantigen. Okay, now second one is isoantigen. These are present only in a subset of the species. Example, regarding the human sapiens or the humans, the blood group antigens and the histocompatibility antigens, they are present only in certain subset of the species. Okay. Then the third one is the heteroantigens. So these are the antigens belonging to two different species. As the name indicates, it's hetero. So these are the antigens which are present in two different species, like antigens of plants, antigens of animals, antigens of microorganisms, etc. Now the last one is a very important one in regarding your exam point of view, that is a heterophile antigen. So this heterophile antigen, it is a type of heteroantigen which is present in two different species, but they share the same epitope. Okay, they share the, so here is the importance, they share the epitope with each other. Okay, they are present in two different species. They are present in two different species, but they share the epitopes with each other. So what does it mean? That is, the antibody produced against the antigen of one species can react with the other, and also vice versa. Did you get me? So heterophile antigens are the type of heteroantigens, which means they belongs to two different species. Okay, and but what is the uniqueness? They share epitope with each other. That is, these antigen can react with the antibody produced against this antigen. Okay, so that is what is regarding the heterophile antigens. Now coming to factors affecting immunogenicity. So let's see what are the factors affecting immunogenicity. So before going to the factors affecting immunogenicity, there are few application of this heterophile antigen. One is, it is used for Weil-Felix reaction. It is also used for diagnosis, and that is the Paul Burnell test, and also cold agglutination test and Streptococcus MG test. Now coming to the factors affecting immunogenicity. So what are the different factors influencing immunogenicity? Okay, we can go one by one. One is the size of the antigen. Larger the size, more potent is the immunogen. Okay, if the size is bigger, that is they can induce immune response. Molecules having a weight of more than 10,000 Dalton molecular weight can induce immune response. So that is, so larger the size, more potent immunogen. Next one is the chemical nature of the antigen. It's another interesting point, that is, proteins are stronger immunogens than carbohydrate, which is followed by lipids, and the least is the nucleic acids. So which antigens will be more potent? That is protein antigen. Proteinaceous antigens will be more immunogen. Okay, immunogenic. Fine. Then next one is susceptibility of the antigens to the tissue enzyme. Third one is susceptibility of the antigens to the tissue enzymes. So what does that mean? That is, only substances that can be metabolized or are susceptible to the action of tissue enzymes are immunogenic. Okay, otherwise if they are not able to be broken down by the tissue enzymes, they are not antigenic. Okay, so only those substances which can be metabolized or are susceptible to the action of tissue enzymes are immunogenic. For example, D amino acids are non-antigenic, while L amino acids are antigenic as they are degraded by tissue enzymes. L enzymes can be degraded by the tissue enzymes. Hence, they are antigenic in nature, whereas D amino acids are not antigenic because they are not susceptible to degradation by the tissue enzymes. Fine. Next point is genetic factor. As you all know, that is due to genetic difference between the individuals, the immunogenicity property of the antigen will vary. Okay. Then next one is the repeated dose of antigen. So before that, regarding the genetic factor, on the basis of the response shown by the individuals, you can classify them as responders. These responders are the individuals who produce antibody faster. Then there are slow responders. And these slow responders are the individuals who produce the antibody slowly, and they may need repeated antigenic exposure. So, and the third one is non-response responders, and these are the individuals who do not produce antibody in spite of repeated antigenic exposure. So now you understood why some that is substances are antigenic to some individuals, whereas some are not antigenic to some individuals, or why some substances produce allergic reaction in some individuals, whereas some it is not showing any allergic reaction to other individuals. Okay, then the next one is repeated dose of antigen. So the next factor affecting immunogenicity is repeated dose of antigen. So over a period of time, the repeated dose of antigen is required, and this is due to the role of memory cells in secondary immune response. Next is foreignness to the host. So what do you mean by foreignness to the host? That is, the higher phylogenetic distance between the host and the antigen, it is higher immunogenicity. Okay, so which means that self-antigens are non-immunogenic, heteroantigens and alloantigens, they are immunogenic. Isoantigen, they are immunogenic to those who does not possess these antigens. So you got it? So when it is in different species, it will be the antigens of different species, it will be highly immunogenic. The next factor which influences the immunogenicity is the optimal dose of an antigen. A too little dose will fail to elicit the immune response, no immune response will be produced, and a too large dose also, that can lead to the development of immunological tolerance. Okay, so optimal dose of antigen is also must for producing an immune response. Now next factor is the route of antigen administration. Immune response is better induced following a parenteral administration of an antigen. Okay, so when you inject the antigen, the immune response will be higher, and also this will decide which type of antibody is being produced. Then the next factor is multiple antigens. Two or more antigens when administered together or simultaneously, there could be different chances. That is, either the antibody production will be enhanced or it will be diminished. Okay, so when multiple antigens are given at a time, the antibody responses to one or the other antigen may be equal or it would be diminished. That could be because of the antigenic competition, or it could be enhanced due to an adjuvant-like action. So what did I say here? An adjuvant-like action. So what does this mean? Adjuvant-like action. So I'll bring your attention towards a term called as adjuvants. So adjuvants are any substances that enhance the immunogenicity of the antigen. So keep in mind, adjuvants are any substances that enhances the immunogenicity of the antigen. So these are usually added to the vaccines to increase its immunogenicity, or the immunogenicity of the vaccine is enhanced by adding such substances which are called as adjuvants. Now I'll give you some examples of the adjuvants. Example is alum, aluminum hydroxide or phosphate, mineral oil, Friend's incomplete and complete adjuvant, lipopolysaccharide fraction of gram-negative bacilli. These are a few well-known adjuvants which are used along with our vaccines so as to increase its immunogenicity. So what does this adjuvant do? They either delay the release of antigen. So adjuvants precipitate the antigen. These adjuvants, they'll precipitate the antigen, leading to a slow release of the antigen. As a result, there will be prolonged antigenic exposure, and they also activate phagocytosis. Larger the size of adjuvants, antigen precipitate increases the phagocytosis. Okay, and also apart from that, they will be activating the T helper cells. This will lead to release of interleukin 11 and express higher level of MHC class 2 molecules, which will further activate the helper cells, and it will also lead to B cell activation and producing specific antibodies. So this is the mechanism of adjuvant action, or the action of adjuvants. Now coming to our third learning objective, that is biological classes of antigens. Now, depending upon the mechanism of inducing the antibody formation, two classes of antigens are there. Mainly two classes of antigens. One more addition is there. I'll explain it to you. So first one is T-dependent antigen. As the name suggests, it is T-dependent. That is T cell dependent. So these are actually the normal antigens. So when a normal antigen comes into your body, what usually happens? You know it very well, right? They will be processed by the phagocytic cells or the macrophages, and they will be presented by the antigen presenting cells, to whom it will be presented to the T cells, which leads to T cell activation. So this is a T cell, right? So this T cell will be activating. Now what does this activated T cell do? The activated T cells will secrete cytokines. Okay, so specific cytokines will be released, and what these cytokines will go and stimulate the B cell to produce what? To produce the antibody. So normal mechanism is the antigens, that is the normal antigens, when they enter into the body, they are processed, that is antigen presenting cells will be processing them, and they'll be presented by the antigen presenting cells or the macrophages to T cell, and you can see it over here. That is, they'll be presented to the T cells through the T cell receptors, and that will lead to the activation of this particular T cell, and the T cell result, the T cell will produce specific cytokines, and these cytokines will stimulate the B cells to produce the antibodies. So these are such types of antigens are called as T-dependent antigen or TD antigens. Next one is T-independent antigen. Now what does this mean? They are independent of T cells. They work independent of T cells, right? So some antigens such as bacterial antigens, that is capsules, flagella, lipopolysaccharides, they do not need the help of T cells or any of the antigen presenting cells. What they will do? They will directly go and bind to the immunoglobulin receptors present on the B cells, and they will stimulate the B cell. Okay, they will be directly going and binding to the immunoglobulin receptors which are present on the B cells, and they will stimulate this particular B cells, and that is polyclonally. This will lead to the secretion of large number of non-specific antibodies. Okay, so that will lead to hypergammaglobulinemia. So T-independent antigens can activate both mature as well as immature B cells. Okay, and the B cells can only differentiate into activated T cells, but there will not be producing what? They will not be producing, they will be producing only antibody, but they will not be producing memory cells. These memory cells will not be produced. Okay. Only IgM antibodies, and mainly IgM antibodies and Ig3 class antibodies will be produced. So this is what is regarding T independent antigen. Remember, they do not depend upon the T cells. They will directly go and bind with the immunoglobulin receptors which are present on the B cells. Okay. As a result, they will stimulate the B cells and they will produce, that is, large number of immunoglobulins, which are actually non-specific antibodies as well. Okay. Next is a very important class of antigen which is called as superantigen. Now, this particular name suggests there is some superpower, right? Okay. So, these antigens, they can activate the T cell directly. Again, they are activating the T cells directly without being processed by antigen presenting cells. So, what they will do? These superantigens, they will come and bind to the receptors. They will come and bind to the receptors, that is particularly the variable beta region. The variable beta region, that is, this is the T cell. So here you can see the T cell receptor, and this is the antigen presenting cell, and here this is the variable beta region, and the superantigen will come and bind over here. Okay, the superantigen, that is the superantigen, it will be coming and binding over here. Okay, and what does it do? So when the superantigen directly bridges non-specifically, again it will come and bind to where the that is between the T cell receptor and the antigen presenting cells, and between the MHC of these cells. Okay, and this will lead to non-specific activation of T cells. Again, the T cells will be non-specifically activated. As a result, there will be massive release of the cytokines. Okay, that will lead to the massive release of cytokines. Again, this will be activating the B cells polyclonally, and that will lead to increased secretion of non-specific antibodies again. Okay, it is not going through the routine process, right? So there is something different happening here. So what is the difference? That is the super antigens. Usually the antigens has to be processed by the antigen presenting cells, and that has to be presented to the T cell receptors. Okay, whereas here what happens is the super antigens will directly go and bind to that is the variable beta region. Okay, between the MHC of the that is variable beta region of the T cell receptor, and they will bridge non-specifically between the MHC class 2 molecule of the antigen presenting cell and the T cells. As a result, the T cells will be non-specifically activated, and that will lead to massive release of cytokines, which will go and activate the B cells polyclonally, and when these B cells will get activated, they will produce they will secrete the non-specific antibodies. Okay, so such class of antigens are called as superantigens. Now, you have also studied certain diseases associated with these superantigens, right? For example, toxic shock syndrome, then food poisoning, staphylococcal food poisoning, then scalded skin syndrome, rare conditions such as atopic dermatitis, then psoriasis, acute disseminated encephalomyelitis, etc. So, this is what is regarding the antigens. Thank you.